So welcome to today's edition of the seminar series on emerging topics in biomolecular magnetic resonance. As you see, we also have a web page where you can look up the upcoming speakers and also directly get a link to the ICMR-BS YouTube channel website to watch the recorded lectures. Just to point out that we follow the Gordon Research Conference policy, so no private audio or video recording of the lectures by any means. And of course, this is also necessary because you will be able to look at them at the ICMR-BS YouTube channel. Just to remind you, if you have any questions either during the talk or after the talk, please use the Q&A button and not the chat version. In addition, you can raise your hand and we can unmute you that you can directly ask your question. So today we have two fantastic speakers. First, we will have Nick Cox from Canberra presenting his latest EPR work. And then in the second part, Kendra Frederick will present her work on DNP, mostly, I guess. And once the Q&A sessions of Kendra is finished, we will close the official part of the edition of today, but then we will stay, at least the panelists and the speakers, to be able to further questions which could not have been answered during the Q&A. So with no further ado, I then would like to start the introduction of Nick Cox, which is not given by myself, but by Gottfried Otting, a colleague from Nick in Australia. So I will play this now.
It is my absolute pleasure to introduce Associate Professor Nick Cox as the speaker of today's magnetic resonance presentation. My name is Gottfried Otting. I'm a colleague of Nick's at the Australian National University, or abbreviated ANU. And while my specialty is NMR spectroscopy, I have learned that pulsed EPR and its applications offer highly complementary and attractive capabilities, as you will hear in Nick's talk today. Nick studied chemistry at the ANU, completing his PhD in 2008. From 2009 to 2015, he worked at the Max Planck Institute for Bioinorganic Chemistry and Chemical Energy Conversion in Mülheim in Germany, under the directorship of Wolfgang Lubitz, first as postdoc, then as group leader. He has won a series of international awards, including the John Y. Young Investigator Award of the International EPR-ESR Society, the Robin Hill Award of the International Society of Photosynthesis Research, and the Paul Saltman Memorial Award Lecture at the 2017 Gordon Research Conference for Metals in Biology. Nick's research has been enviably fruitful, with a range of articles in Science, Nature, and PNAS just in the past 3 years. So we were lucky to be able to attract him back to the ANU in 2016 on a future fellowship by the Australian Research Council. Wolfgang Lubitz referred to Nick as, I quote, a very gifted spectroscopist who strongly contributed to the development of instrumentation and methodology. I myself noted that he is also a very popular lecturer of undergraduate classes in chemistry. Without further ado, I'm clearing the stage for today's lecture by Nick Cox. Enjoy.
Thank you, Gottfried, for the nice introduction. And now I have to again share the screen to play the pre-recorded lecture.
Hello. Well, I'd like to thank the organisers, Christian, May, Marcus, for the opportunity to present an overview of our research on nature's water-splitting reaction developed using magnetic resonance, specifically electron paramagnetic resonance and related double resonance techniques. But before I do, I just want to briefly tell you about the EPR facility we're setting up at ANU. So I'm joining you today from Canberra, Australia's national capital. This is the Parliament. And if you were to turn your head to the right, you would see the ANU campus, with the Research School of Chemistry being just here. And it's here we're setting up a new EPR facility, a modified commercial platform with the help of the Australian government and my old pals at the Max Planck Institute for Chemical Energy Conversion. It hosts Australia's only high-field W-band EPR instrument, along with more standard X and Q-band instruments. It's set up as a flexible platform allowing in-situ illumination, electrochemistry, gas exchange, etc. If you're interested in anything I show today or have a pressing EPR project, please contact me. We'd always be happy to help. With that short preamble, I'd like to turn to the topic of my talk, biological water oxidation, which is performed by a single enzyme in nature, photosystem II. This is a topic of interest in the field of renewable energy as it provides a blueprint as to how to make renewable fuel, say hydrogen, cheaply from abundant materials, sunlight and water. The water splitting reaction is catalyzed by a pentaoxygen tetramanganese calcium cluster, for which there are now high-resolution crystal structures. It adopts a distorted chair motif. I shaded the back of the chair green, with the chair lying on its side. It's located on the luminal side of the transmembrane protein. All manganese is 6 coordinate, except for manganese 1. So there's an open coordination site within the cubane unit. That's what I'll call the base of the chair. How it works? Well, photosystem II is like nature's photovoltaic. Light excitation of the multichlorophyll assembly leads to a charge-separated state, allowing sequential one-electron oxidations of the manganese cluster. It stores the oxidizing equivalents needed for the water-splitting reaction. After 4 light flashes, 4 oxidations, the manganese cluster then splits 2 water molecules into molecular oxygen, returning to its resting state. So O2 evolution has a 4-flash response. Thus, the catalytic cycle of the manganese cluster can be described in terms of 5 states, 5 S states, with each state representing a 1-electron oxidation of the cofactor, making it particularly well suited to study by EPR. Note that the S1 state, not S0, is the dark stable intermediate. So the X-ray structure I showed you on the previous slide was the cofactor poised in the S1 state. What I want to show you today, essentially I'll cut straight to the end of my talk, is an almost complete reaction cycle for the water splitting cofactor developed from magnetic resonance spectroscopies. Note, in this diagram I've rotated the cofactor 90 degrees such that the back of the chair is now in the plane of the board. Within this cycle, the cofactor moves from a resting type structure to an actuated form with a new water molecule inserted into the cubane unit. The O bond formation then likely occurs between these 2 oxygen ligands. The 2 key stages of the cycle that magnetic resonance informs on are the dynamics of the cofactor in allowing the second substrate water to bind, and the electronic configuration of the final intermediate, which likely promotes a radical coupling mechanism as described by Per Ziegbahn. It's these 2 points I'll elaborate on in the second part of my talk. But before we get there, we first need to quickly cover how to view the cofactor using EPR. In EPR, you measure unpaired electrons, which the manganese ions, in either the +3 or +4 oxidation state, all carry. They're all local high spin. But you don't see 4 EPR signals associated with each manganese. You see only 1. And that's because the electrons on adjacent manganese sites couple together via superexchange mechanism. They align either anti-parallel, anti-ferromagnetically couple, or parallel, they ferromagnetically couple. The important thing is that these interactions are mediated by the electronic overlap between the d orbitals of the metals with the non-magnetic oxygens of the bridge. As such, how the manganese couple together is intrinsically related to the structure of the complex, and thus the overall spin state of the complex can be used as a marker for structural change. The magnetism of an exchange coupled system can then be described in terms of a ladder of effective spin states, of which we're usually only sensitive to the ground spin state, as this is the only one populated at cryogenic temperatures. Note, transition metal complexes usually have fast magnetic relaxation, forcing us to measure at liquid helium temperatures. For a multi-spin system, such as the manganese cluster described here, it's the complete set of couplings that then describe the ground spin state. Here I show you the exchange coupling pathways for an early intermediate, early S state of the cycle, the S2 state. As the dominant interaction is antiferromagnetic, the ground spin state is low spin. It's an effective S equals 1/2 state, with the state, if you like, having one effective unpaired electron. I'll refer to these 2D representations of the cofactor as a coupling map or a coupling topology. They're essentially how EPR spectroscopists view the structure of the cofactor. Turns out that all early S states adopt the same low spin topology, S0, S1, and S2, which implies that they all have the same geometry. A distorted chair, open cubane motif, what I showed you earlier. The EPR spectrum of the S2 state looks like this. Its spectrum, the multi-line spectrum, is centered around G equals 2, typical of an S equals 1/2 species, which is then split due to the hyperfine interaction between the effective unpaired electron, carried by the whole complex, and the 4 manganese nuclei. The interpretation of this hyperfine structure is ambiguous, so we turn to methods like ENDOR, in which we measure the NR&R spectrum of the complex using the electron spin as a reporter. I'll show you these types of measurements for the next intermediate of the cycle. Okay, so moving past this point in the cycle, the magnetism of the cofactor dramatically changes. The exchange pathways within the cofactor switch from being dominantly antiferromagnetic to dominantly ferromagnetic, leading the cofactor to adopt a high spin ground state. What this means is that the cofactor structure has fundamentally changed. The EPR spectrum of the S3 state is a bit more complicated than that of the S2 state, as it's an integer spin system and a non-Kramers state. I don't want to go into a long discussion of the EPR of high-spin systems, if but to say that such a system will generally have a very broad EPR signal that's made up of multiple allowed EPR transitions which overlap, as shown here, analogous to quadrupole nuclei in solid-state NMR. How we assign this spectrum is to first turn to model complexes. Turns out that the EPR spectrum of the S3 state is very similar to simpler biomimetic cubane models in which you only have octahedral manganese ions, suggesting the cofactor has lost its open coordination site, its 5-coordinate manganese ion. This comparison is based on the total width of the signal. Put simply, manganese ions with an octahedral ligand field tend to display a small zero field splitting. And it's this magnetic property, the net zero field splittings of all 4 manganese ions, that controls the overall width of the signal. That's what breaks the degeneracy of the MS transitions of the ground state spin manifold. NMR, ENDOR type measurements, support this assignment, where we now measure the NMR transitions of the 4 55 manganese nuclei via the electron spin. Remember that all the experiments that I will show you today in this talk are performed in the solid state, and thus the EPR spectrum represents the cofactor in all possible orientations. By then measuring the NMR spectrum at different positions across the EPR spectrum, we can then selectively look at only one orientation of the cofactor. Note too that we get a different NMR spectrum depending on which EPR signal we use as the observer. As the NMR signals of the 4 manganese ions don't change, look say at the edges of the EPR spectrum, where we're using the blue EPR signal to observe our NMR spectra. There is no orientation dependence. And as such, the electronic structure of all 4 manganese sites must be locally symmetric, a property of octahedral manganese 4 ions owing to their half-filled T2G levels. So the spin state changes because the stoichiometry of the cofactor changes. An additional water ligand is bound to the cofactor at the open coordination site, closing the cubane unit, the base of the chair. So an understanding of spin state evolution during the cycle is telling us something fundamental about the cycle. It's telling us the transition when the second substrate binds to the cofactor. In the last few minutes of this talk, I'll show you that we can go further and actually pull apart precisely how this occurs. If we turn back to the S2 state, what I didn't mention earlier is that under certain circumstances, it exhibits a second EPR signal characteristic of a high-spin species. Now, these two forms of the cofactor are interconvertible, even at low temperatures, meaning that the stoichiometry of the cofactor is not changing, as we saw with S3. So what is instead happening here? Well, from what I showed you previously, you would expect that one of these signals, the multi-line signal, represents the open cubane structure, which promotes antiferromagnetic couplings, while the other high-spin signal presumably represents a closed cubane structure, which promotes ferromagnetic couplings. Well, my colleagues in Mulheim developed a model for how this could be possible. It's called the interconvertible structures model, in which one of the oxygen bridges, the O5 ligand, can toggle between two positions. This motion is coupled to, it's driven by valence isomerism, the position of the only manganese 3 ion of the S2 state, which changes from within the cubane unit to the outer manganese. Although I don't have time to talk to you to tell you about it in this talk, we've also studied binding of the two substrates to the cofactor via O17 labeling, using similar double resonance methods. And these data definitively show that O5 is one of the substrates of the reaction. We have a paper on this hopefully coming out soon. Now, we'd argue that it's this dynamic nature of the cofactor that's critical to its function. What it does is it moves the open coordination side of the cofactor of the cubane unit to the more solvently exposed outer manganese, thus promoting second substrate binding through either the aspartate 61 channel, its water channel leading to the cofactor, or the second water channel leading to the cofactor, the calcium channel. Our most recent study provides further evidence for substrate binding occurring in this fashion. In this paper, we identified now a second form of the S3 state. It's best seen by adding small molecules like methanol, which are known to interact with the water channels of the protein leading to the cofactor, and thus likely slows or competes with substrate binding. What happens when you add methanol is you see this much broader form of S3, requiring the cofactor to still have a 5-coordinate manganese ion. That's to say the second substrate water is not yet bound. 55 manganese NMR data support this conclusion and are consistent with its structure resembling a closed cubane similar to the preceding high-spin S2 state. Thus, the sequence of events becomes clear. Cofactor breathing, this movement of the O5 ligand, allows second substrate binding to the cofactor. But this only happens after cofactor oxidation. This makes good chemical sense. Water binds not to a manganese 3 ion in the S2 state, but instead after the manganese 3 ion is oxidized to the +4 level, the manganese 4 center being beta-Lewis acid. Putting this all together, starting off in the lowest S state, the S0 state, the cofactor can progress to the S1 state by oxidation of the back manganese, manganese 3. We then progress to the S2 state by either oxidation of manganese 1 or manganese 4, the interconvertible structures, which allow water to access the open coordination site of the cofactor. Cofactor oxidation then leads to the first S3 intermediate, which following a water binding and rearrangement gives the final S3 intermediate structure. It's this final S3 intermediate following a further oxidation event that can catalyze OO bond formation. But I hope you instead come away from my talk thinking about the cycle like this, as composed of two discrete phases corresponding to the resting and activated forms of the cofactor. And it's these two forms of the cofactor that correlate with a low and high spin coupling topology, respectively. What I want to close with is just a brief explanation as to how the emergence of a high spin topology likely promotes low barrier OO bond formation. Per Zygmunt first proposed that the OO bond formation likely occurs through a radical oxo-oxyl coupling. In this mechanism, the last oxidation is ligand-centered, forming an oxyl radical within the cubane. The coupling topology determined for all the high spin states, which I showed you here, supports this mechanism as it ensures that all subsequent manganese-oxygen bond breaking events, which occur commensurate with OO bond formation, lead to the 3 manganese ions which bind the product O2, gaining an electron of the same spin as their existing d electrons, which is to say that all of these electron transfer events occur without energy penalty due to spin conversion. It also explains why the product is a triplet O2. That said, I should emphasize that what I've shown you today doesn't prove this mechanism. The best you can ever do is disprove the mechanism. And part of our current work is trying to trap the oxyl intermediate invoked in this mechanism and characterize it via 17O labeling, as we've done for the substrate sites earlier in the reaction cycle. With that, I'd like to thank everybody involved in the project, which has spanned over 10 years now, especially my team back in MPI, CEC, and my new group at ANU, and the many collaborators as well. And thank you again for having me, and I'll be happy to take questions.
Thank you, Nick. So now you can take questions.
Right. So there's no questions in the box.
Always takes a bit.
Okay. All right.
So I have a question for Nick, why don't you move from W band up to say 140 or 250 gigahertz to do the indoor experiments? Is it simply a matter of the availability, or is there a fundamental reason that W band might be better? I mean, you've gotten a lot of information from the indoor.
Sure. No, there's no. I could see advantages of going to higher frequencies, certainly with the resolution of some of the manganese couplings that we see in the final S3 state, which are intrinsically small and thus somewhat overlap with the blind spot that you see in the indoor. Strictly speaking, we're not measuring these via indoor, we're measuring them via LDOR detected NMR, ED-NMR. So we have the blind spot issue to cope with. I sort of brushed over all of that. T1 relaxation is a big problem here. So avoiding the indoor experiment and moving to the ED-NMR experiment is ideal. So yes, indeed. Certainly, that I can see advantages going to higher frequencies.
And why didn't you use some 017 labeling?
We did.
Or did I miss that? Was I asleep?
Sorry. So we've done all of the 017 labeling experiments as well. I just didn't have an opportunity to show that. But these allow us to detect the substrate sites as the two substrates exchange rapidly with bulk solvent. So in that way, we can discriminate them from structural oxygens. And what we've also been able to do is correlate the induction of the 017 signals we measure with mass spec measurements, in which the rate at which the substrate exchanges can be measured. In that way, we can make a definitive assignment as to which 017 signal corresponds to a substrate and which one doesn't.
Mm-hmm. Yeah. Thank you.
So there's two questions here. So the first question is from Marina Benatti. How can you conclude from manganese hyperfine on structure? So in the talk, I just emphasized on the emphasis was on the local properties. So we were simply trying to infer what the local ligand field of the manganese were by their field dependence, their orientation dependence. And the absence of this shows that the four manganese signals are likely to be spherically symmetric. So they're likely to be 6-coordinate octahedral manganese ions. There's another aspect to this, though. We can also measure the spin densities across the whole cofactor via our manganese ED-NMR indoor type measurements. And it's this disbalance or whatnot that allows us to make more rigorous structural models. And this is a process that involves computational chemistry, so DFT type calculations. So there's a second sort of element to this that I didn't get time to talk about in the talk, in which we can actually refine structural models based on these experimental constraints. Okay, second question is from Arthur Palmer. Being ignorant about details of EPR, in the alternate S2 structure at G4, are the hyperfine couplings broadened to be unresolvable, or is there something else going on? Yes, typically in powder spectra, you don't resolve the manganese hyperfine couplings. So you also don't see them, say, for the high spin S3 signal either. they are indeed smaller than the line width. However, you can see them in orientated samples. That has been done. I have never been able to get a pulse DPR experiment to work on the high spin S2 state. Its T1 relaxation is just too fast. So which precludes sort of a more in-depth analysis of the hyperfine couplings of this state. But as I say, there is some data in the literature, originally from the Brit laboratory, which definitively shows that that G4 signal does have a hyperfine structure if you measure orientated samples. Question from Dalia Avila. Do you perform this measurement at low temperatures? Yes, all measurements that I showed you in this talk are all performed at low temperatures. So the manganese cofactor has very fast T1 relaxations of the order of hundreds of nanoseconds at low temperature. And that already makes a lot of double resonant experiments like Endor already very challenging. It's unfortunately unavoidable. Okay, I think
Thank you, Nick.
no more questions.
Thank you, Marcus. So the next speaker today in the emerging topics in biomagnetic resonance is Kendra Frederick. Kendra was an undergraduate at the University of Michigan in Ann Arbor, where she got a degree in biochemistry and I think also in French and maybe something else as well. She then did a master's degree of sorts at the University of Paris, where she again was working in a biochemistry laboratory, I believe on SUP35, a prion type protein. She then returned to the United States and the University of Pennsylvania, where she did her PhD with Dr. Josh Wan, very famous NMR spectroscopist, who's done a lot of really beautiful solution NMR experiments, and that was in the Department of Biochemistry and Molecular Biophysics. And for a postdoc, Kendra took a big leap in a different direction. She came to MIT, to the Whitehead Institute, to a yeast genetics lab, which at the time was run by Susan Lindquist. And she spent a good deal of time there working on various and sundry projects. And at the time, same time, she was sort of honorary member of the Griffin group, where she started learning about something called dynamic nuclear polarization, right? DNP, and that doesn't mean do not publish, it means dynamic nuclear polarization. And she's taken another big leap now and tried something very hard with DNP, which she's going to be telling us about today. She's been doing all of this work at the University of Texas Southwestern Met Center in Dallas, Texas. And the title of her talk is In-Cell Structural Biology Enabled by DNP Mass NMR. So Kendrick, the virtual floor is yours. You unmute yourself.
Hold on. Do this.
Okay. You're muted again.
Yeah, I think we're having dueling muting buttons. It's great.
We can hear you now.
Excellent. Yeah. Thanks, Lauren. All right. So it's really exciting to be able to give a webinar here today. And thank you for the invitation. I like to start my talks by showing this plate of yeast because from my time in the yeast genetics lab, this is the type of problem that has really caught my attention and forms the center of the biological problems that my group is working on. And so on this plate here, every single yeast you see is genetically identical, yet they have clearly different phenotypes, right? And these phenotypes are stable right? The beginning of the streak is the same as the end of the streak in color. And if I were to plate these yeasts on a different plate where they had different food, this would be their choice between life and death, right? So it's not just a beautiful change in phenotype, it can also be a deadly one. And in this particular case, it's all genetically identical. The difference between these yeasts is the conformation of a single protein. And so I'm really interested in how protein conformation can affect cellular fate. And this isn't just a problem in yeast. It's one that we know very well in the biomedical community. And that's in the context of neurodegenerative disease. So in many neurodegenerative diseases, there's proteins that have one conformation at the beginning of the lifespan, and in the absence of genetic changes, take on a different conformation towards the end of someone's lifespan and have a big effect on phenotype, have a big effect on life course, right? And so shown here, immunohistochemical sections of diseased brains where the misfolded or the protein that changed conformation is highlighted in brown. And one thing that's really terrible about these diseases is although we can treat the phenotype, there's not a single therapeutic strategy that aims at the underlying protein pathology, right? And so I think that one of the issues that we have here is that we're actually, as structural biologists, not particularly sure what the toxic conformations or the toxic folding pathology really is. And I think that that sort of comes to this idea of protein folding and misfolding. So for a lot of biochemistry and structural biology, you have a polypeptide, it comes off the ribosome, it's unfolded, makes an intermediate, makes a native state. And for much of structural biology, particularly crystallography, for example, we are pretty sure that this conformation, this native state, corresponds to what you have in the cell because they have shared aspects like protein kinetics or the mechanism that they act by. But for these other proteins, clearly the environment intervenes, changes the size and the types of these arrows, and you end up in different spaces. And so for these protein folding diseases, the environment is really critical. And getting that environment correct is probably also critical to understanding what the toxic conformation might be. Right. And so what we really want to do is study protein structure in a complicated environment. But you really want to just see a single protein and have everything else there to make sure that you have the right conformation. Right. And so, of course, many people try, like you can use light microscopy and get an idea of morphology. Of course, the resolution is limited there. You can get beautiful structures from cryo-EM. You can purify fibrils, for example, out of diseased brains and get structures there. But of course, you're selecting for a subset of the ensemble. You don't have everything. Right? You can do cryo and electron tomography, and that means every single thing is there. But of course, you're limited in the resolution. And in order to get any sort of structural information out, you have to average a bunch of objects. And so things that have different conformations are more difficult to parse. So of course, spectroscopy, NMR is by definition an atomic technique, and we all know that there's beautiful specificity through using the correct isotopes, right? And so then it is uniquely suited for these types of problems, right? And so, you know, a lot of this work has been started in solution state NMR simply because of the sensitivity. But there, of course, you also have the selection bias because you only see the things that are tumbling quickly. And for these complicated systems where you have proteins that make complexes or make fibrils or have large correlation times, you really need solid state NMR spectroscopy to get the entire ensemble. Okay. And so today I want to talk to you about the work that my group is doing towards a system for in vivo structural biology enabled by DNP and MAS NMR. Right. And the reason that we want to do this is to maintain the biological integrity of the sample in order to understand which conformations are represented in health and in disease. And so there's a couple of challenges, of course, with this. Sensitivity is something that we're going to address with dynamic nuclear polarization because that's a sensitivity enhancement technique for solid state NMR. Specificity, of course, we know that we can isotopically label our protein and introduce it, and we stand on the shoulders of giants from the solution state NMR spectroscopy community for techniques for introducing specifically labeled proteins into cells. And then resolution, which I'll touch on briefly, but won't expand upon here. So I want to just go back to this briefly and talk just briefly about the protein that changes conformation here on this plate. And that protein is sub35. It's the translation termination factor in yeast. And it has 3 domains. It has this domain here that's called the preend domain with the N-terminal piece and this middle piece, which is highly charged. And then it has this C-terminal domain right here that's involved in translation termination. And so in a yeast that has an artificially introduced stop codon in the adenine synthesis pathway, what happens is the ribosome goes along the message, gets to this artificial stop codon, doesn't finish making the protein, and you get a buildup of this red pigment. But of course, if you take this sub35 right here, right, it's involved in translation termination, and you basically deplete it from the cytoplasm, and you make these big aggregates, then the ribosome gets to the stop codon, can't stop, reads through, and you synthesize the adenine correctly. You don't get a buildup of this dye. Right? Now, this doesn't happen, so you could keep making these aggregates, but of course this is a heritable phenotype, something that keeps going. And so you have this conformation, this aggregated conformation. You can pull normal protein in, and then things inside of the cell, like chaperones, are involved in breaking this piece up to pass it on. So this is an inherited conformation. And so this is largely done in the context of yeast through the chaperone called HSP104. And if you genetically delete HSP104, you end up with large aggregates and you don't get inheritance because they're sequestered in the mother cell. If you overexpress HSP104, it works too much and it breaks down these aggregates down to their original tiny pieces. And so you also lose inheritance of this conformation. And this works in collaboration with a variety of other chaperones in the cell. If you overexpress or delete them, you also change the inheritance patterns of these aggregates. And so that means that the concentrations and the stoichiometry of this stuff in the cellular environment is critical to maintaining this conformation and propagating it and its biological activity. Right? So there's lots of molecular insights into the NM preends, and purifying conformations. We know that this N-terminal domain is really rich in Q's and N's, forms amyloids, and is absolutely required to make those aggregates. This M domain is rich in highly charged residues. We know that it binds to chaperones. It's intrinsically disordered in purified systems, and it's absolutely required for prion inheritance. Right? And so the question is there any difference between the types of conformations we can propagate in a test tube versus types of conformations we can propagate in cells? Right? And so the approach is clear. Right? We want to isotopically label this NM protein and have it and assemble it into these fibers in the cellular environment. Right? And so what we do there is we take these yeast, we grow them up in isotopically depleted media, we lyse the cells open, we add isotopically enriched NM protein, and then we can watch the appearance of fibers over time when we add small amounts of that protein to the lysates. Right? And so this is a Western to show that that's true. We can add protein at very low concentrations. This is approximately 1 micromolar. The native concentration is 2.5 to 5 micromolar, so we're not really pushing the stoichiometry. Right? So the added proteins at the endogenous conformation or concentration, and we also have tests to know that the added protein is in the fibril form. Right? So that's how we make this sample. Now, of course, that's a terrible NMR sample, this one right here, because of the sensitivity. Right? And we all know this, and perhaps this is even unnecessary for this audience, but that's because, of course, like a very, very small amount is polarized at a really high magnetic fields for carbon. Right? And that's defined by this energy splitting right here, gyromagnetic ratio, and the strength of your magnet. Right? So if we go from something with a lower gyromagnetic ratio to a higher gyromagnetic ratio, and then we transfer polarizations using a cross-polarization mechanism, we can increase the sensitivity of the experiment. But if we take the really high polarization of the unpaired electron, we heard about this just a minute ago, we can take that polarization, and this is dynamic nuclear polarization, we can drop it all the way down into the nuclei that we want to see. Now, it's not perfectly efficient, but we have the possibility of just ginormous signal enhancements here, right? So we take this type of sample, we add a source of unpaired electrons. So this is a stable bionitroxide radical called amipole, right? And then we put it in the magnet, and we turn the microwaves on, right? In order to do the frequency matching, in order to transfer polarization to the protons and then to the carbons. And what we see when we do that is the DMP works in complex biological mixtures, and it works actually very, very well, right? We get enhancements of about 100, and that means that one experiment with DMP is equal to 100 experiments without, right? So the experiment in 2 dimensions is like 10,000 times faster, and that's completely enabling. We can now think about looking at tiny, tiny amounts of isotopically labeled protein in these complex environments with the rest of the stuff is there, but we just don't see it. Okay, and so now the question is, does the environment influence the structure? Right, so I'm showing you here the carbonyl carbon chemical shifts for a purified preparation of these fibrils, right, just in buffer, and then these fibrils that have been prepared inside of the cell lysates. And if you look at what the chemical shift distribution should be, particularly the chemical shifts that you would expect if it was in an alpha helical form, you see that there's a bunch of contribution from this purified sample in the alpha helical form, and that's completely depleted when this same protein is put in lysates, right? So you get a huge influence of the environment on the secondary structure. And if you look at different regions of the spectrum, you can actually localize that change in structure to a particular region, because while there's Q's and N's right here, and there's a lot of them, I actually can't tell you with the resolution that we have in this spectrum anything about what their conformations might be. But in this intrinsically disordered region that's lysine rich, we see shifts of more than 4 ppm. Right? So the environment influences the structure of the region that's disordered in purified samples. Okay, and so that means that biological contexts can and do influence protein structure. Now, the challenge, of course, is resolution, not sensitivity or specificity. I want to give you just a little bit of insight into how we're very, very sure that we're totally okay for sensitivity and specificity. And so this is the primary sequence of that protein that I told you about, and this is a spectra of 1 micromolar of that protein in about 30 milligrams of cell lysates. And what you can see is in this 2D, this is where the lysines are, this is where the arginines are, this is the proline and the tryptophan. If you compress all that all the way down into a skyline projection, if you look at a projection, you can see that we detect signal for arginines, just these 2 arginines right here. And we don't see any tryptophans. This is what would be in the background cellular matrix because there's no tryptophan in the protein. We have more signal from proline and lysine. So we have the sensitivity and the specificity to be able to detect a protein at 1 micromolar in these backgrounds. And we can detect 2 amino acids. So what we're doing for resolution is to label just small portions of the protein in order to cut the problem down to size. So this is that same protein sequence right here. If we label every single amino acid with carbon and nitrogen, this is what the glycine region might look like for 1 fibril type. But if we label just this first segment and you only have these 5 glycines, you cut the problem down so you have these tiny peaks instead of this whole mix. Okay, so I'm not going to talk about that today, but just to say that this is the approach that we're taking to get the resolution we need. So I really want to talk about in vivo structural biology, and the reason we want to do that, of course, is to maintain the biological integrity. And with DMP and MR, we have the sensitivity, specificity, and resolution to do it now. Okay? And the reason we want to do this is because we want to take cells, understand their phenotype, do an experiment, get a structure or structural ensemble, and then make sure that we didn't perturb anything at the end of the experiment, because there's these big questions about what the toxic conformation is. And so if we take cells and we do that with a gradient of drug that changes the phenotype, we can then see how that gradient affects the structural ensemble in our experiment, and then make sure that we haven't messed anything up at the end. Same idea with genetic changes in the genetic background.